Our next speaker is Fabian Theis. Fabian comes to us from the Institute of Computational Biology in Munich, where he is the director of the institute, correct? And he's also a coordinator of a network here in Germany for single-cell omics. And we welcome you very much into this session. Thank you.
Yeah, thanks for kind invitation and for the possibility to be here. This has been fantastic. Thanks, Genevieve, Nikolaus. The place, the people, the discussions, I think is going to be a lot of fun. Although you tasked us with a bit of a challenge, we should not only talk about our own research, but we should try to sort of put things into context. So I try to do this in the next 15 minutes or so. So I want to speak about lineage inference, and I call this latent spaces in single-cell genomics. That should sort of indicate a subspace of a high-dimensional gene expression space. And I would argue that the lineage that we infer is nothing but a tree latent space that we actually try to learn to our large-scale data. However, I want to start with the latent space that, Nikolaus, you always put so fancily on those beautiful images that you make. So this is my latent space that I want to project now your cell trajectory on and learn it from that. So this is basically what I spent most of my time preparing my slides on. Let's go back to the usual thing. So I want to speak about single-cell RNA-seq, and this session is called data science and, was it data science, machine learning, and AI, so you got all the buzzwords covered, right? So I think it kind of fits because over time, single-cell RNA-seq has been actually turning into a large-scale data science problem. I think Amos so nicely put it that for 5 million people, you can put it on a memory stick. If that was to be single-cell RNA-seq data, it might not be the case. So obviously, people have realized this, that at some point it might be actually necessary to talk about that. It used to be called boringly statistics. Since then, there have been some reviews even in science that speak about still a bit black and white, machine learning and AI. And I think it actually has some meaning in our context because it has revolutionized a series of disciplines. If we look back what has been done in computer vision 5 years ago, this has completely changed. Everyone's using deep learning, similar in natural language processing. And I like to argue that the reason why this is the case is that nowadays you have so much data in those fields, nicely annotated data, that we can actually learn representations of the data now in much better fashion than we would just naturally do it. If anyone remembers drag and dictate from 15 years ago with this manual sequencing of vowels, that was not as good as if you learn it. So similar, we try to do something like this feature representation, for example, and this is basically what those two fields are now using by deep learning, where you feed in some type of non-nicely separated data. At some point in your layered architecture, it becomes nicely separable and you do some interpretation. And I'd like to show 2 examples that this is also happening or starting to happen in our field. I'll be mostly focusing on transcriptome data, but clearly there's multi-omics integration, and I think there's also a strong link to spatial techniques. What's the biggest challenge in the field? Let me give you one more intro slide, because we were supposed to be generic. We had a recent single-cell omics meeting. We asked the audience what they think that big topics are, and what came out was, again, always this data integration, multi-omics integration aspects. And what I want to show in the following is that this latent space learning, in fact, has data integration as a special case. So I think if you're good at determining a latent space in one experiment, you can actually do this by stitching together multiples as well. Let me start with the single tree or even linear latent spaces. Those are just lineages or pseudotimes or trajectories. So we have a bunch of cells in a high-dimensional transcript space. They hopefully don't form just a big old blob, but they lie maybe in something that has a lower dimensionality than the 10,000 dimensional transcripts or so that we have. And this space we could call a latent space. It might be curved, nonlinear. If everything was linear, we'd be done with PCA, but every one of us knows that this is not the case. And therein, we might, as a first approximation, order things linearly. If you want to be fancy, you could call this a geodesic distance in this sort of differential manifold, but we call it plain old pseudo-temporal ordering, and there was some insight to be gained from that. And in fact, a lot of computational scientists, us included, have picked that up as a fun discipline. And I think in this review, there was already, I think 40 or so different algorithms reviewed. Recent count was up to 79. So it has become a fun discipline to come up with good orderings for that. In this nice review from Nir Yosef and Aviv, they actually tried to piece together other sources of variation that went beyond only this single linear architecture. For example, they spoke about discrete cell types, continuous ones, some sort of branched direction, cell cycle, or something spatial. All of these are implementation of a topology, in this case a discrete one. So if we look for clusters, we search for sort of separate blobs in our latent space, if you want. In this case, a line, a tree, circular, complex. So one idea, and I think that's the goal of what people call manifold learning and machine learning, or in our case, I guess, latent spaces, would be maybe to do something uniformly. And one object that in the computational field has been very, I think, common to work on, I think since the first, I guess it was phenograph, that was one of the first papers to be actually using that from Dana Peer, where is to use it, look at a single cell graph, which is just a KNN graph, just connect cells by similarity of the expression profiles, or if you do multi-omics, some other type of profiles. And then one approach that we did, this was worked by Alex in the lab, was to do something called partition-based graph abstraction, to now do some on different level of coarse graining, and then be able to connect those clusters, in our case, not only with a tree, but with a graph to also capture the uncertainty. I don't want to be only fluffy, so I want to have one content slide before going to the more algorithmic one again. So we actually apply this, and we have some also local first nice own data. This is a collaboration with the Lickert lab, where we've been interested in mouse gut development and establishment. In this case, also then in a challenge study on metabolism. But here I just show the normal data. We are asking, is there a single unipotent progenitor state, or does it go via multipotent states in between? So we're asking, do we see direct differentiation or sort of intermediates? For this, we do single-cell and seq. There was some nice enrichment tricks that had to be done, but in the end, we end up with our clusters that all of us have seen so often, and then we superimpose this structure of this graph abstraction that I showed you before. Then we see that our intestinal stem cells are actually connected to multiple progenitor states, but no multipotent ones before we go in the target ones, so we could actually invalidate that from what we had at the moment. But I think what was really nice that then Heiko came up with an actually labeling strategy of the stem cells to see then full lineage tracing, how this was going. I think there's much more fancy, or sort of there's more elaborate techniques now that the colleagues have been speaking about now. Let me do one slide on a concept that you might not have heard so often about. You know, we speak always about manifolds, but in fact there's a hardcore definition of what a manifold is. And that sort of illustrates this thing here. In manifolds, for some reason, people always speak about donuts because, well, I guess they like donuts. But this is maybe also interesting because it has sort of a non-trivial global structure, but an interesting local structure. So latent space is nothing more than just a subspace that looks like some linear space locally everywhere, but could be globally complex. How do you describe this mathematically? Well, what you do is actually you say that there's in all kinds of neighborhoods, you can always make it flat with some type of map. If you do this for multiple neighborhoods, then you always can make them flat, but you also know how to deal with those intersections. Those you wouldn't be able to map between those, and that's how you define then additional properties to the manifold. So what I'm trying to say is, locally we know what to do, and this is like a globally nonlinear PCA, which locally gives you something nice, but globally does you strange things. Very often, if this was to be our atlas of, let's say, the gut, as I showed you just before, we would be doing some type of t-SNE or UMAP of this whole thing, maybe arbitrarily cut it up there, because obviously you can't make a donut flat, unless you eat it or something. But in this case, you have to do something different. So locally, you can do some analysis, but globally not. What are the applications of that? Well, so far I showed you that we could, of course, do lineages, right? So we could now say that piecing it together, this would be part of a line, this would be part of a line, so we would be sort of piecing it across there, or if the donut was branching off, we'd be able to do that. But in addition, instead of just stitching one data set together, local linear, we can also do this for multiple data sets. And in fact, this has been something that I showed you in the beginning, the state integration being a central task of many current research in the field. So for the stitching together, you would be doing the same. So this could be data set A, this could be data set B, where you have an overlap of some cell types, but you would be maybe then more generally doing something. And I think, and that's why I wanted to at least put it out here as a suggestion, you know, we should be coming up with new ideas what we want to do computation in the long run in this lifetime project. One idea would be, is there some global properties of that object that we're trying to estimate that could maybe be describing something more than just clusters, essentially? And there's ideas from the field that you know, for example, counting holes in these things, or seeing what's map invariant, what's these measure actually true geodesic measures on that thing. There's different ideas for that. This has been realized, of course, by the field already some time ago. I think there were two seminal papers from John and Raoul's lab that have both been dealing back to back, in fact, with batch correction methods that were exactly going along this line of piecing things together. In this case, with this beautiful idea of mutual nearest neighbors, where they've been essentially describing a graph structure on both levels of batches and then have been sort of merging those graph structures. And Raoul has been taking this up in his most recent paper with anchors, sort of extending this towards integrating multiple omics techniques. There's a similar paper from Peter Kachenko's lab where he's doing this on a large scale across many different labs. So these are all graph-based techniques, and we contributed also a test then for seeing how good those batch integration methods work. So all of these are techniques that essentially use this single-cell graph and extend it to doing this map-based stitching together that I showed you before. You could, however, argue that you might be interested in doing more than just graphs. For example, you want to extrapolate. Say if in our case we have some diseases that we won't be able to actually see in one particular setting. We want to be able to extrapolate, to put two things together and go completely out of sample. For this, it might be interesting to know first which genes of our transcripts actually contributing to which area in our thing. So this map specifically see where's the map, its derivative changing most strongly or something like that. This goes in the direction of explainable AI, which essentially asks those questions to deep neural networks. Or we would be interested to see how those multiple of those genes actually interplay to give us one particular coordinate. And this is with these graph-based techniques maybe not as straightforward to do. So what I tried to suggest, and I think some people, in particular initially near Joseph and others have been picking this up for some time, to use something that the other fields, and initially I showed you audio and speech processing, have been doing for some time, namely using deep neural networks as a tool to learn strongly nonlinear latent spaces. In our field, mostly using unsupervised techniques. We take essentially a transcript space as input, squeeze it down, then blow it up again and make the output as similar as possible to the input. If you do that, you find essentially a latent bottleneck encoding that if everything was linear would exactly reflect a PCA, in this case could be doing something more. If you do this, for example, in this particular example, you would be reconstructing this latent space that somehow illustrates this progenitor differentiation. There's other ideas, a recent paper from Gramani et al. That uses something called generative adversarial learning, do some training, but those things are a bit of a beast and don't yet converge as nicely, but the idea of generative modeling is around, and I encourage you to read up on that if that's of interest. Won't be speaking about that any further today. Why is there, though, no supervised learning? All of this is unsupervised. The big advantage why I think 2012 or so ImageNet was coming out and those image processing field completely changed to neural networks was because they had this large-scale annotated image database. In our case, we could ask, well, couldn't we do prediction of, let's say, cell type annotation? That is a nice predictive learning task. So what we actually did, we sat together, we looked into, in this case, 4 different studies on human pancreas and tried to look at this learning annotation problem. In this case, this works beautifully well, so we just leave out one study and predict on trained of the other studies. In this case, we leave out this parent study and predict now cell type from the actual known cell type in that study, and it seems to be working out very well using our neural network. But if we now compare this, and now here you just have one prediction, now we just change false positive to positive rays, look at this area on the curve. And if you compare this now, our neural network with gradient boosting support vector machines, or even just a simple old linear method, turns out that they all actually perform pretty much the same. Let's do this in a more extended study. So we go up to smaller as well as much larger studies, and we see that except for SCMap, where they didn't do some sort of systematic parameter screening, all of them perform pretty much the same. This tells us that in fact the annotation, cell annotation task is just too simple a task in contrast to, for example, finding cats in images, that you actually don't need deep learning for that. So the learned filters, the learned latent space representation here is not interesting enough. So we might need more functional screens, something like that, to actually really get full deep learning to be working and be actually feasible in that area. So let's go back to the latent space learning. What could you do, however? So what really got me interested was not so much this denoising technique, but that this bottleneck, in fact, actually does represent something. So in this case, I show you PBMC data set that has been annotated, as we usually do, look in clusters, enriching for cells, and then just reduced to a two-dimensional bottleneck name. What the two-dimensional bottleneck more or less learns is something that looks pretty similar to a t-SNE or UMAP. So it actually separates the clusters. So this means that those squeezing down, blowing up things do learn something about the latent space. Not perfect, but these latent space seem to be presenting something. So what we asked, can we now predict also a perturbation? This is our goal in lifetime. We have diseases mapped on top of that. Can we predict perturbation effects on those simple latent spaces? what we set out, this was work by a PhD in the lab, Muhammad, and together with Alex, to now look at our gene expression space, learn for a set of cell types where we know perturbation effect already. This might be already where we have set up our disease atlas. Now we have a new one coming in. We try to predict the effect of that disease. We do this encoding with the autoencoder that I showed you before. And interestingly now, in the latent space, things seem to be separating. So we can actually show in quite a series of examples across different species even, that those latent spaces turn out to be rather linear. So we can actually learn this linear translation and then apply it to this new unknown cell type, project it back, and then learn this type of predicted cell type. This is what we call SCGen. It uses, again, a generative model, this SCVI-based variational autoencoder, and do this out-of-sample prediction. And essentially, if you apply this to PBMC data where it's stimulated under control, we leave out one sample, and then we can essentially predict the stimulus just as well as the real stimulus, not only across one gene, but across the whole transcriptome. So this was rather fascinating to us. There seems to be something that this autoencoder has learned about some type of epigenetic state, and now you can start interpreting things. So as an outlook, what do you do with this? Well, obviously, you can now do something like experimental design. You could propose potentially new optimal type of perturbations, if that works. You can use this very efficiently for batch effect correction or do some style transfer, essentially learn on one species even, go and transfer to other. All of this, obviously, with a need for much better latent noise models. And you can do this trajectory inference as well. I think caveats are that those things are more or less still black boxes. They sum up across multiple layers gene expressions. So doing this interpretation is a challenge, but I think the AI field is currently moving strongly towards more explainable AI methods, and I think we could harvest and make use of those methods in our field as well. And of course, once this works out, you could think about this might be our HCA, where we have studied the unperturbed situation rather well. Well, this is kind of what we try to do in lifetime, right? And there's so many perturbations, filling up those perturbations might be a bit of a chore. So maybe we can harvest and use those computational methods to fill this up more efficiently. I'd just like to point out that the imaging field is already doing this in parts. For example, the Carpenter Lab at the Broad is using a technique called cell painting, where she just labels with a few markers and uses then morphometry that she measured in another situation to then impute additional phenotypes also on her data. So this is my conclusion. I think I tried to show you that those latent spaces kind of can be used in general to describe variation across data. You can understand topology, and you can go beyond a single experiment by doing multi-data set integration. Current techniques are graph-based, neural network-based. It's not so easy to fuse, actually, the two approaches, at least to my knowledge. But downstream, you could go towards more interpretation or even more mechanistic modeling. This is my lab. I think I already acknowledged everyone doing the work here, and I thank you so much for attention.
Thank you, Fabian. We got a question here.
So, super interesting as always. Now, here's a kind of conceptual question. So, I think it's very clear. So, you show very nicely how far you can go with dealing with the cell trajectories, in essence, as some kind of input-output problem. Okay. And that is hard enough and difficult enough, and it starts to work in many aspects, and there's super nice work and emerging work and so on. But of course, you can also say, ask a question, is it really possible, how far can you really push this without incorporating molecular mechanisms? So, of course, you know, if you think about the trajectory, the differential can be explained by molecular mechanisms, and also if you perturb them, then this will define the change in trajectory. So, do you think you can actually do without the molecules? You know, I would have thought that what is really necessary in the long run is an integration of this. So that in the end, if we think about how to predict, there should be equations or operators which should reflect the molecular machinery. But maybe you say we do it like in the social sciences where Google predicts what we buy next, and that's already very powerful.
This is a great question, kind of connected to what Amos also initially said, that we sort of want to fill up this error from prediction with actual cause and effect. So I have essentially two answers to that. The first one is, that I'm quite convinced that if we do more and more of those data sets, that at some point we can maybe even understand what is a lab difference effect, and we could maybe do all kinds of predictions, maybe even learn something about the regulations even in those simple superposition things. I do think that in images, for example, it has turned out initially not to be a good idea to describe what a face is by saying it's composed of nose, ear, and whatever, but actually let those representations be automatically learned from the data. However, as soon as you want to actually then do something with that, in our case, for example, in diseases, there's rare examples where we never see some realization of that. We have to go in data, in areas of our transcriptome space or of our spatial, whatever space we want to extend this to, where there's areas we won't be able to do this prediction. For those extrapolations, I'm pretty convinced that more mechanistic approaches would give you a much more secure way of generalizing your predictions. Having said that, in this particular example with the PBMC example where we left out the CD4 positive T cells, we actually tried to do this. We left out more and more cell types and said at what point did it actually break down. And we saw that at some point it does, and I think there's some uncertainty model to put on top of that. But I think the future, and this is essentially this whole area of what people call explainable AI, is nothing more than going to worse prediction models than the optimal one, but doing some type of additional constraint, which in our case could be something mechanistic. So I think this traditional area of what we did in systems biology 10 years ago, so remember, where we had all those equation models that were not so predictive in many situations. I think this could be taken up again now and included. And there's some first approaches, a technique called physics-inspired neural networks that has actually a neural network representation of the ODEs, and because of this beautiful TensorFlow framework, you can actually optimize them fast. So there's basically people putting things together from both areas. So I think, in particular with new measurements that say if you give us now some additional genetic labeling, some real true velocity and so on, those things could be coming together.
Thank you. One last question, if possible, shorter and shorter answer too.
Sorry.
Yeah, so I'll just follow up on what Nicolaus was asking, because there's anyway some overlap. So what do you think is the sort of roadmap of going from these sort of latent spaces whose coordinates are really abstract to getting to coordinates that have direct physical biological meaning, so that we may actually relate coordinates in these spaces to underlying molecular mechanisms?
Thank you so much for asking that. So this concept of the donut that I showed you, it means that you do have, for each location, you can make multiple maps. And in those first batch correction techniques that we had, always had to have one reference data set that we corrected everything on top of. What I try to argue with this donut type of thing, and some of the recent techniques are doing that, you actually represent your latent space in some abstract coordinates, but you can always project in your own lab's coordinate or someone else's lab's coordinate. Similar to language translation, people nowadays do learn not only across 2 languages, but across 20 languages at the same time, and then they can actually show that the learned filter is getting better than just learning on 2 languages, and they find something latent that's arbitrary coordinates, but you can always project in the coordinate framework that you're interested in. So I would hope that this is also possible at some time point. For transcriptomics, it is already. I guess if you have imaging and so on, and that's also this question I asked this morning, right, where you want to understand what's a generic representation of an image. I think it's not a good idea to make one standard model that is taken in your lab, but it should be translatable.